engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
Discoveries.
Advances.
Questions. Research.
technology. Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists podcast, the show that brings you the biggest breakthroughs and talks to the major movers and shakers in the worlds of science, technology and medicine. I'm Chris Smith, and today we hear from Titan of Science, Sara Wickstrom, who has pioneered how cells sense and react to pressure and other stimuli and then store their experiences in their DNA. So does this mean that individual cells can have a memory? Clap your hands, stamp your feet, or pull on a pair of shoes that are too tight, and in the days to weeks that follow, your skin cells in the affected areas will respond to those shockwaves by toughening up. They'll lay down more protective tissue, but they might even go a step further and store a memory of that insult in their DNA. The implications for cells sensing, remembering and responding to pressure and other stimuli speak for themselves. Think about a developing embryo where cells jostle for position and rely on knowing who their neighbours are, so all our body parts ultimately form in the right places and with the right proportions. Or consider how cancers behave when this system goes wrong and grow invasively and without any regard for normal tissue boundaries. This is a crucial branch of biology, and the person who's pioneering a path to understand how it works and to harness it to make us healthier is the scientist and doctor Sara Wickstrom. Born on the 30th of April 1976 in Espoo on the outskirts of Finland's capital Helsinki, she's now the director of the Max Planck Institute for Molecular Biomedicine in Münster, Germany. And she's just won the €1 million Corba European Science Prize for her discoveries. She's our Titan of Science this week.
One of the tissues that we study is the skin, or the epithelials, or the surface part of the skin. And it's kind of clear that although the skin is composed of the exact same cells in the different parts of our body, but the composition in the end is very different. If you think about the eyelid, think about the elbow or the sole of the feet, the kind of the consistency and the composition structure is very different. So this already made me think immediately that there must be some mechanics involved, how much pressure or how much stretching the tissue experiences, and maybe that kind of guides the functional specialization, and that's how I kind of got into the subject.
So in the same way, if I wear a pair of too tight shoes, initially I might get a blister, but 2 weeks later I've got much thicker skin there and I don't get a blister again. The skin somehow knows, in inverted commas, the pressure effects have changed and it needs to change.
Exactly. It first senses and then it adapts. And that's essentially what we study, how the sensation happens, how the adaptation happens, and whether sometimes the adaptation is not good for the tissue in the end.
Is that at a local level? Is that decision-making cells locally doing that? Or is there a more central mechanism that says, hey, nerve cells here are getting wound up, let's send a message back to tell the skin to change? Which is it, or is it both?
That's a really great question, and we don't exactly know, so that's still something that we're actively investigating. Does every cell sense on its own and make its own decision, or is there some collectivity involved, just as groups of cells, or we even across cell types like epithelial cells and neurons? So that's still an open question.
How might cells, if it was just cells on their own, do it, though?
Well, we know, for example, that much of the sensing is happening at the cell surface, so the plasma membrane. So if you imagine that the cell is experiencing stretch, there are so-called ion channels that gate how ions flow from outside of the cell inside. And some of them are mechanosensitive, so they might be, for example, opened up in response to the stretch, which generates ion gradients that trigger changes in cells.
And okay, that would change the electrical activity that the cell is feeling, but crucial to that is responding in the right way. So how does that happen?
Right. So one important mechanism, and that's what we have discovered and we study, is that how gene expression changes in response to, for example, stretch. And what we have learned that these ions, but also other signals, are actually relayed to the nucleus of the cell where the DNA is stored and regulated in a protein complex called chromatin, and how these ions and other signals actually change the chromatin and thereby change which genes are being expressed. And that directly then changes how the the cell will behave.
Presumably different cell types are sensitive to different sorts of pressures then. You've talked about skin and we walk on that, we hold things with our fingers, etc. But there are going to be other cells in other parts of the body that are experiencing quite different stresses, but it would be useful for them to sense those.
Exactly. So that's what we think is part of the adaptation. If we think about development, we know that actually forces are important for the development of the cardiovascular system. So the shear flows from the blood or the stretching and contraction of the muscle will actually further guide the specialization of the tissue. And then these similar forces that might cause massive changes in skin cells no longer change the behavior of these cells because they are adapted to experience these continuous forces. But on the other hand, they might actually react differently when these forces stop. So this means that the cells are specialized to sense their force environment, and what we think they're doing is that then they are actually they are sensing the change, and that might trigger these rapid responses in gene expression that we see.
Presumably, although we think we're very special as humans, we're obviously evolved from a more simple or more primitive biology. Presumably then this is much more widespread than just us.
Absolutely. I think these are conserved mechanisms, and we already know that at the origin of multicellularity is actually compressive forces that trigger the cells, single cells, to attach tightly to each other. So I think these are universal mechanisms that have evolved for organisms to sense their environment.
You see, when I first read about your work, I was reminded of one of the first interviews I ever did when I was making science programs, because we were very lucky to speak to one of the team at NASA who had unfortunately recently lost Columbia, the space shuttle. But one of the experiments that was aboard that was coming back from space where they'd flown moss in microgravity. And the question they were asking is, well, would it grow? And what was amazing was A, the experiment survived the crash and they got it back and they were able to analyse it, but B, when they analysed it, and this is about 20 years ago, they saw these bizarre spiral growth patterns. And when we interviewed Volker Kern, who was one of the contributors to that study, I said to him, well, why would the moss know it was in space if all other things were equal? And they said, well, what we suspect is that the moss cells can detect gravity. And they speculated at the time that there were particles of starch and things inside the moss cells that were settling under gravity on Earth and bending the inside of the cell. And that's how it knew what was up and what was down.
Yeah, that's really fascinating. And I also think that cells are being able to sense microgravity. And that's, for example, how balance is being sensed in the inner ear. What's interesting, though, is that it's probably based on scale. So, for example, it's known that certain cells are very large, and there probably microgravity plays a more important role because of the structures being sensitive to gravity, versus smaller cells might not be so sensitive. So there might be interesting special aspects of size that are related to sensing microgravity. But that's a very difficult topic to study. Not all of us can send our samples to space.
No, indeed. But there must be situations where if you can unlock or work out what the genetic programs are that are interpreting these signals, it might have really important clinical applications. Because I'm thinking, if we think cancer grows invasively, the cells don't know where to stop, is one of the reasons why cells normally know where to stop, because they feel the pressure from adjacent cells and that switches them off, and cancer's ignoring that.
Absolutely. I think there's two things that are likely important for cancer. One is indeed that they might become insensitive to these kind of signals that normally, for example, restrict growth, but they also might in fact become more reprogrammed, because if the force environment changes and that changes gene expression, that actually might also lead the cells to differentiate into different trajectories. And that's what we have also shown, is that cancer cells, when they experience pressure that is derived from the growth of the tumor, they actually might gain properties that make them even more aggressive.
So they subverted the system and sort of flipped it round. So what would normally be a handbrake now becomes an accelerator.
Exactly.
Wow. And do you know what genes it invokes to do that? Because obviously if we want to try and gain or regain control, knowing how it is doing that is critical.
So what we have learned is that it's maybe not one or two key genes. It's more essentially the entire cellular program, because I already mentioned chromatin, because all of our cells have identical DNA, and the chromatin is there. So the protein component that packs the DNA is there to control which genes are expressed in which cells, and that makes them specialized. And now what happens in the case of pressure, for example, that the entire chromatin structure kind of changes, that allows more genes to be accessible for activating factors. So it's more like an entire reprogramming of the genome than switching on one or two genes.
So it sort of unwinds the DNA into a state that can be read, or winds it into a state that can't be read. It's pretty gross rather than discrete individual genes.
Exactly. And that's why it's maybe harder also to target with drugs, because it's an entire reprogramming that, rather than activation of a pathway that could be targeted with drugs.
But why would we go for such a brute force mechanism rather than a more discrete? If a cell can sense some pressure, turn on this suite of genes, or turn on this control gene that then controls other genes.
I think initially it might be beneficial for the cell to do this in a healthy situation, because these kind of mechanical stresses are maybe something that is evoking these adaptations, and that's why in these stressful situations it might be beneficial for the cell to react very fast. So by changing the chromatin structure, it becomes very efficient for the cell to launch these kind of adaptation mechanisms that we discussed. But now in disease, these stressful situations become prolonged, and maybe then these adaptive responses are not launched in time, and thereby the cell has kind of a longer period of time to launch more kind of malignant or less beneficial adaptations. This is more speculation at the moment, but if we compare the signatures of healthy and cancer cells to these stresses, on short time scales they are very similar, suggesting that it's not like a pathological response, but rather a healthy adaptation response. But somehow these stressful situations seem to last longer in disease, maybe kind of prolonging the window of opportunity for the cell to then receive additional signals and become more and more altered.
When people train, they're obviously putting huge pressure on different tissues. Is that adaptive, as in, are they plugging into the system that you work on, and that's how a muscle knows, apart from it's being injured a bit, because obviously when we work out we do injure our tissues a bit, but is that part and parcel of how tissues think, I need to get bigger because I've just had a workout?
Yes, so basically when we look at these gene expression changes that I mentioned, a major component of genes that are being changed are these so-called cytoskeletal genes, which are the components of the contractile machinery that the muscle uses to generate force. So exactly this adaptation and the ability to generate more force, but also resist deformation. These are kind of the same machinery is used for both, and that's why the cell, by increasing the production of components of this machinery, will be able to sustain its shape and maintain its function in response to, and in the presence of this kind of mechanical stresses.
When we develop as embryos, because obviously we all come from a single cell that then divides and divides and divides and turns into 37 trillion cells in an adult human, all in the right place, amazingly, in the majority of us. Do these effects play out there too? Because that ball of cells, there must be quite a bit of contact and pressure signalling going on for things to then know, right, okay, I'm in the middle, or I'm at the edge, and I have to become skin, or I have to become a heart.
Yes, so that's what we're actively studying. So we're kind of working out the mechanisms, but we exactly believe that the forces, although they are not the only players in development, obviously, but they are kind of guiding to help and reinforce the signaling decisions by exactly giving this kind of positional information. But it also might be that this mechanosensitivity is something that develops during development, and there might be periods of time, for example, in very early development, where the cells are less sensitive to forces. But this is something that we are studying at the moment.
And do any microorganisms also plug into this? Because we've talked so far about health, we've talked about development, we've talked about pathological states and also reactive states, like me wearing too tight of shoes. But there are things like parasitic infections, malaria, for example, that need to know about the environment they're in to react accordingly. Malaria glues red blood cells to blood vessel walls, for example, to stop the spleen grabbing them. Does this play into that? Or is there any evidence that microorganisms and parasites might be subverting this same system?
Yeah, so there are some really exciting studies, for example, from bacteria and biofilms, and how actually bacteria also sense mechanical forces and use it for infectivity. So definitely this is not something that is restricted to eukaryotes or mammals.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
This is the Naked Scientist podcast with me, Chris Smith, and I'm talking with Titan of Science, Sara Wickstrom, about the discoveries that she has made about how cells sense what is around them. Now, you've won a pretty significant prize for unlocking the door and opening the door on a lot of this and really shining a light on this world. What actually is that award, and what are you going to do with it?
Well, it's called the Kerber European Science Award. So this is an award given every other year to a bioscientist and every other year to a physicist or a computational scientist. And this is a award of 1 million euros that one can use for research. So it's not going on my private account. It's going to my research account.
It's a shame. I was going to ask you to buy me a beer, but
yeah, I can still do that, but not with the million. So the fun thing about this award is that it's still flexible to use, because normally in science we are very constrained with our grants in terms of budget, how much we can spend to hiring students, how much we can spend on equipment, and how much we can spend on consumables. But this grant is more free to use, and this means that more room is given to serendipity, creativity, and just kind of the scientific progress. And the focus of the research that I will use this grant for is to try to understand whether these effects of forces on chromatin and gene expression is something that the cells and the tissues can remember. So as an example, the cell has experienced mechanical stress, but then the stress goes away, but whether some traces of memory will be retained in the nucleus, in the chromatin, that when the cell gets another similar experience or even something a little bit different, it will behave differently because it's had this previous experience, similar to us. So essentially, it had made an experience and it recalls this experience that changes its future behavior.
What a fascinating idea, because I first came across this about 20 years ago when researchers began to point to the link between obesity and the fact that a person pregnant and starved had children that were much more likely to turn into diabetics who were obese and with high blood pressure later. The Dutch hunger winter was used as an example, and they were able to point to epigenetic changes in certain genes that had occurred to cause that. So that was a biochemical stress. So you're sort of saying that physical stress might be remembered in a similar way in cells then?
Exactly. So there are some hints that this might be the case, but I think it has not really been stringently and rigorously tested. So that's what we plan to do. And particularly we're interested in whether pathological tissue states, so whether if a tissue has been injured, whether that could leave these kind of memory scars, and when then another injury or another insult arrives, whether this previous injury is somehow affecting how the tissue will respond to the next one.
For instance, if you had an episode of extremely high blood pressure and put massive load on your heart, would your heart cells remember that they were under more stress and therefore permanently or semi-permanently label some genes that might then affect, say, disease risk or even resilience in future? Exactly.
I think both effects are possible. It could be adaptive and thereby maybe beneficial because the tissue is more resilient, or it could be maladaptive and render the tissue more sensitive to the insult. So both outcomes to us are equally interesting and equally plausible.
Are you going to look at the level of individual genes with this, though, or back to what you were saying earlier about the fact that it's whole chunks of how the DNA is wound up and put into an inaccessible or a more accessible state that you think underpins a lot of this?
Well, we think that the initial response, and we know that the initial response is quite global, it's still possible that there could be certain memory genes or certain genes that are more prone to be part of such a memory, and they might be the important genes that then regulate the future responses. So here we are definitely also focusing on individual genes, because these, of course, could act as fantastic markers for such memories, that we could detect them in tissues and also trace them over longer periods of time to kind of try to understand this behavior also in the context of tissues and not just in single cells.
Well, I'm thinking, because someone once asked me, if you have a heart transplant, does the heart have any memories from the person it was in that's then conveyed to the new recipient? And I sort of laughed at the time and said, well, no, that's not how memory works. But actually, you're sort of rewriting that book then, because if the heart from the donor has got one of these sort of epigenetic memories in it now of past pressure experiences, it could well carry some change in function into that new body.
Well, in theory, yes. And I think our research should be able to find those genes and understand whether it's relevant for the future health of this transplant recipient.
Do you know how to rewrite the book, though? So although you can see changes when tissues are subject to pressures or stress or being bent and deformed and distorted, and this translates into genetic changes, is it a one-way street, or do you think you can rewrite the book?
Well, that's, of course, the ultimate goal, especially if it turns out that these memories are disadvantaging the tissue. So then, of course, it would be amazing. And of course, if it's encoded in chromatin, we know already a little bit based on other people's work how to modify, for example, these chromatin readers or writers. So in theory, it might be possible to rewrite the memories, and that, of course, is a dream outcome of this research.
There are some drugs that, as side effects, do this kind of thing, though, and we discovered this as a sort of byproduct of looking at these drugs. We actually invent them for something else. They treat the condition they're designed to treat quite well, but then you find, they also do things to the chromatin. Are there any drugs that serendipitously happen to do this, and which can give you some insights into how this might be working?
Well, there's an important class of drugs called the bromodomain protein inhibitors, which are used for cancer, and they are exactly targeting these epigenetic enzymes. So they could be a good candidate. Of course, the problem with these drugs is that they are, again, targeting a very broad range of chromatin effects. So again, whether they could achieve the position of then controlling these few genes that are important, that, of course, is something that is likely to be challenging.
Now, when you first started out, you actually went to medical school to start with. Did you actually become a doctor for a while, or was science just too much of a lure and you got seduced by that and didn't practice?
Well, I did, but actually I went to med school, but I always wanted to be a scientist. I just didn't, because I don't come from a science family, but I wanted to become a scientist, but I didn't understand how I will achieve that goal. So I went to med school because they had the MD PhD program, which was appealing to me, because then I thought, okay, maybe that's my path to science. So I went through med school, but I already started also doing cell biology in medical school and fell in love with cell biology. But then after I got my MD and my license to practice, I then finished my PhD. And after my PhD, I worked 1 year as a general practitioner, so just in a healthcare center, seeing patients, just to be sure that when I'm as old as I am now, I will not have a midlife crisis, and thought that I should have become a clinician. It was a really rewarding experience. I enjoyed actually being able to help people, but my love of science was greater. So after this 1 year stint, I did my postdoc, and after that, I have never returned to the clinics.
And what was the journey between Finland and where you are now?
It was a journey of serendipitous decisions and kind of just always thinking about the path to do the best possible science. So I ended up going to Germany kind of based on just the lab. So I was not thinking about moving to Germany to be in that country. So that was a decision driven by science. And then I got an offer to be a group leader. So I ended up staying. But then in 2018, I got an offer to become an associate professor in Helsinki, which was my hometown. So I went back to Finland and stayed there for 4.5 years before then the Max Planck offered me this director position, and then I decided to return to Germany.
They realised what they'd lost.
Who knows?
But what do you do when you're not in the lab? Because, lots of scientists do interesting, exciting things away from the bench, which is where they have their thinking time and they come up with their best ideas when they're not trying to think of them, sort of reverse psychology. So where do you have your best ideas?
Well, I'm thinking about science a lot in random places, but I do love sports and exercise. So I do some road biking, some running, and all kinds of outdoorsy stuff. And usually I end up thinking about science at some point when I'm enjoying getting some exercise and some fresh air.
I know you're thinking about all the pressure effects on your cells when you're doing that.
I try not to think about that though. Yeah, so it's more important to just enjoy the movement than to think about mechanical stresses. Indeed.
But if this comes off and you're able to get evidence that tissues do have these sorts of pressure memories in that way, what will this unlock, and where will you take that next if you're successful?
I see two possible trajectories. One is that we find memory genes that we can use as markers, because my lab is also very interested in understanding how the tissue architecture and how the shapes as proxies for these forces, because it's very difficult to measure forces, for example, in diseased humans. So we rely on using shapes and architectures as proxies. And it has turned out that we can actually develop those analyses into diagnostic tools for cancer. And now I envision that if we can find these memory genes, we could also use them as biomarkers that could tell us about the disease progression. So that's one application where we would like to take this. And the second is trying to erase or write these memories in tissues. For example, fibrosis is one important health threat. So actually, it's estimated that 50% of deaths actually result from fibrosis, either as a secondary outcome of heart infections or infectious diseases or cancer. So if we could somehow tackle these fibrotic reactions by rewriting these mechanical memories, so the cell, instead of producing more and more of this very stiff scar tissue that prevents these normal organ functions, it would start remodeling and basically restoring the normal tissue stiffness and architecture.
Absolutely fascinating. Sara Wickstrom there, Director of the Max Planck Institute for Molecular Biomedicine in Münster, Germany, and winner of this year's Corbo European Science Prize for the work that she's just been explaining. Will transplanted organs turn out to have a memory of what they've been through? Wouldn't that be an extraordinary finding? We wish her all the best of luck. Now, we're back with the latest science news stories of the week on Friday, of course, including this one we just sniffed out, that a baby's brain synchronises its activity with that of another person when they can smell their dad. We'll discover why scientists think that is. Meanwhile, if you'd like to get in touch, you can drop me a line via chris@thenakedscientist.com, and do, please, if you have a chance, also drop us a rating and a review wherever you get your podcasts to help us to spread the word. Lastly, if you appreciate what we do here for you each week and you'd like to support the show, do please consider making a donation. You can do so at nakedscientist.com/donate. And to those of you who already do, thank you so very much. I'm Chris Smith. From all of us here on the team, thank you for your time and for listening to us, and until next time, goodbye.